hopefully get people starting to come in. Hopefully your talk is a little less than an hour, so we have time for questions at the end.
It's going to be 45 minutes or so, but I can adjust
that's great.
As we go.
No,
yeah,
that's all good. Give it just a minute for letting the stragglers find their way in. So as people come in, I do want to first thank you all for being here for the Quantitative Systems Biology Seminar Series, and make a comment about the next seminar in 2 weeks on February 10th, Wednesday, February 10th. We have a different time for our seminar. It'll be at 9:30 a.m. We have a special speaker coming in, and that is the only time they had available. So we're going to hopefully make sure everyone is aware that the seminar will be starting early at 9:30 in a couple weeks on Wednesday the 10th. We will, of course, send out more information to people's emails, but there we go. So today, I have the pleasure of introducing Mohammad Fallahi Sichani from the University of Virginia. I've known Mohammad for a while now, and I'm really looking forward to hearing his talk, but let me give you a bit of a background on Mohammad. He originally got his Bachelor's of Science and Master's of Science in Biotechnology from the University of Tehran in 2007, and then came to the United States and the University of Michigan to get his Master's of Science in 2009 and his PhD in 2012 in chemical engineering underneath the supervision of Jennifer Linderman and Denise Kirschner, both amazing modelers in their own right that acted as fantastic mentors for him, where he developed the tools to build multi-scale models and applied those to investigate inflammatory signaling mechanisms in tuberculosis infection. In 2012, he went on to Harvard to do his postdoctoral work in the laboratory in systems biology in the laboratory of Peter Sorger at Harvard Medical School. And during that time, he was awarded this highly prestigious Life Sciences Research Foundation postdoctoral fellowship. It's a very highly competitive postdoctoral award. So that was an amazing feat. And after his postdoctoral work in the Sorger lab, in 2017, he joined the faculty at University of Virginia, where he is now as an assistant professor of biomedical engineering. He's since been able to get a whole bunch of funding from the NCI as a K99 award, from the V Foundation for Cancer Research, from the Department of Defense, a Career Development Award, and a Maximizing Investigators Research Award from the National Institute of General Medicine. So again, it is my pleasure to introduce our speaker today, Mohammad Fallahi Sichani. Thank you.
Real quick, if I can interrupt. Hi, everyone. We're going to do things a little bit differently this time. So as you have questions throughout the talk, please feel free to drop them in the Q&A box. And Mohammad will field questions at the very end. I'll read them aloud, and we'll just do all the Q&A at the very end. Thanks.
Thank you so much, Darren and Kavya. It's a great pleasure to be here. Thank you so much for the invitation. I'm very happy to be virtually presenting the work we've been doing in the past few years on systems pharmacology approaches that we use to better understand the mechanisms of heterogeneity in cancer therapeutic responses. So I want to start with thanking everyone who have contributed to the project that I'm going to talk about today, especially Mehwish, my amazing postdoctoral fellow, and Mohan, who have led the majority of experiments that basically I'm going to talk about, my mentors during the past years, and the funding sources. So I'm going to talk today about cancer targeted therapies. These are treatments that, as you know, are motivated based on dependency of tumor cells on mutated oncogenes and activated pathways. So it is based on analysis of genetic mutations in driver oncogenes across patients that are basically allowing us to predict what sorts of treatments are used for basically therapy of cancer in different patients. Examples of these are, for example, BRAFIMEK targeted therapies, EGFR targeted therapies, and other targeted therapies that are currently used in the clinic or under clinical development for a variety of cancers with different driver oncogenes. So I want to focus on the BRAF mutation, which is the focus of our studies that I'm going to talk about in the context of melanoma. BRAF is a kinase involved in the RAS/MEK/ERK signaling pathway that, in the presence of growth factor, can transmit the signals to the cell to initiate normal growth and survival. But what happens when BRAF is mutated at the V600 position, the protomer of BRAF becomes constitutively active. It doesn't require RAS signaling or dimerization to induce activation of ERK signaling pathway. And that is what's happening in a variety of cancers, especially in more than 50% of melanomas that patients are observed with BRAF V600E mutations. So the good news is that we have BRAF/MEK targeted therapies developed in the past 10 years that can control the activity of signaling pathways, slow down the growth in melanoma cells, induce death, and basically they are currently used as one of the treatments available to BRAF mutant melanoma patients. But the problem that many people in the context of a variety of cancers are working and we're interested in the same context in the case of BRAF mutant melanoma is the heterogeneity in drug response. What we're observing here is the responses of melanoma patients, 132 of them, as evaluated by percent change in tumor size in response to BRAF inhibitor vemurafenib. And pretty much amazing observation is that we see a full spectrum of drug response, almost linear observation that basically we see every type of response that is possible, from full responses to no responses, with the majority of patients be categorized in a group that we call partial response or residual disease. And the problem is that in the majority of those residual disease cases, ultimately, most of the patients acquire resistance and develop their tumors back. And these tumors are resistant to BRAF/MEK targeted therapies. So there have been a variety of studies focusing on finding ways to overcome drug resistance in the past 10 years. And now we can agree on a model that's basically happening that describes the behavior of cells during response of them to BRAF/MEK target therapies. And that response is grouped in two subgroups. One is the short-term responses that happens within the first few days and weeks of treatment. And as you see here, because of the BRAF mutation and oncogene dependency, melanoma cells are dependent on this pathway, so they stop proliferating or die. But at the same time, some of the tumor cells acquire ways to adapt to treatment and tolerate the effect of drug. And as we keep cells under drug treatment for longer than that, additional epigenetic or genetic changes are acquired by these cells, and they become fully resistant and ultimately associated with tumor relapse in patients. So there have been many studies. I'm just citing a few of them here in this slide, highlighting mechanisms that are involved in either short-term adaptive responses or long-term acquired resistance mechanisms. And there have been solutions proposed to overcome drug resistance. And one of the ways that multiple groups, including us, have been working on is that can we, instead of waiting for these tumors to become fully resistant, develop strategies to maximize response of tumor cells in the first place, so we have a smaller niche of cells and we have a reduction in the probability of acquired resistance. So that requires that we understand the origins of heterogeneity in drug response in the first place and finding ways to overcome those mechanisms and maximize drug effect. So I'm going to talk about this problem. I'm going to show an example of drug response that is happening in Colate58 cells, one of the BRAF-mutant melanoma cell lines that we use in our lab. And this study basically shows that by profiling drug response in time, we reduce basically the growth of melanoma cells in a dose-dependent way, but even close to highest concentrations of bemrafenib above the IC50 of this drug for these sensitive melanoma cell lines, we reach a plateau in drug response, which means that some cells survive the treatment and we're not able to eradicate them by even increasing the concentration of bemrafenib. So this somehow highlights the importance of live cell analysis or analyzing the growth rate instead of basically looking into snapshots of drug response. And because of that, we have profiled the response of a variety of BRAF-mutant melanoma cell lines to get a better understanding of the types of responses that we see. So these are responses of 16 BRAF-mutant melanoma cell lines to either bemrafenib alone or bemrafenib in combination with trametinib in comparison with primary melanocytes. And as you see, we see a diversity in heterogeneity of response similar to what we are observing in patients. Every possible response is basically observed among very dramatic responses, intermediate responses, and no responses in the same BRAF mutant melanoma cell lines. So to understand these heterogeneities at a systematic level, we are interested in quantifying these, and we use the same strategy that have been used by multiple labs in Vanderbilt, and basically use drug-induced proliferation rates based on the normalized growth rate of melanoma cells in the presence of drug compared to untreated cells or DMSO-treated cells. So that allows us to quantify the effect of drug on melanoma cells and compare them across different cell lines, whether they are not responding or they're showing some cytostatic behavior or cytotoxic behavior. So by doing this strategy across different melanoma cell lines, we can calculate the net growth rates and then basically normalize it to the DMSO-treated sample. And based on that, we can summarize the comparison between response of different melanoma cell lines. So LOX-IMBI cell line, for example, do not respond at all. We have cells like SKML28, which shows cytostatic responses, and there are cell lines that are showing a lot of cytotoxicity. So in order to understand the reasons behind these heterogeneities at a single cell level, we have engineered these cells with reporters that can help us understand what is actually happening in melanoma cells at a single cell level. And these reporters can help us track changes in the activity of ERK signaling, which is the target pathway for bemrafenib and trametinib. And basically tracking cells can help us understand whether cells survive or die, and also cell cycle progression. All three of them at the same time using a triple reporter. For ERK activity dynamics, we use the ERK-KTR fused to the mTOR-CoACE reporter. And for tracking the nuclei and cell survival and death, we use the H2B-Venus reporter. And for tracking cell cycle progression, we use an mCherry-Geminin reporter. So by combining these, for example, we can track the behavior of individual cells in the presence or absence of drug. This is, for example, showing the response of individual melanoma cell without any treatment. As you see here, tracking an individual cell shows us the cell division time, and basically tracking Geminin can help us understand whether cells are in different phases of the cell cycle. They are typically showing low levels of Geminin in the G0, G1 phase, and during the cell cycle progression, the Geminin reporter shows accumulation. And at the M phase, which is mitosis and division of cell, the Geminin reports reduction, a rapid reduction in the levels of this report. So we can compare between individual cells in the absence or in the presence of drug. As you see here, for example, the vemurafenib slows down the growth of an individual cell, but and the cell that I'm showing here is dividing almost 3 times more slowly than a drug-naive or DMSO-treated cell. So to get a collective behavior of single cells at a population level, we can combine tracks of individual cells in the absence of drug or in the presence of drug. For example, here, this is the COLID58 that I showed the population behavior before. In the absence of drug or presence of DMSO, cells are dividing almost every 24 hours. So these black dots are division events, and wherever you see a line, a yellow line means cells are alive, and transition from yellow to gray is actually cell death. So by comparing the drug-treated cells, you can see that in the first 24 hours of treatment, the majority of cells stop dividing, and then a fraction of them die. And among the surviving fraction, about a half goes back to proliferation and keep dividing, but slowly. And another half is remaining in a quiescent state. And this is the response of cells within 84 hours. If you extend this time, you're going to see that there are slowly cycling cells that are almost dividing every 72 to 80 hours. And there are some cells that do not divide even after after a week or 10 days. So combining these data with the levels of ERK activity and Geminin intensity, we can also see whether cells are reactivating the ERK signaling. And that shows actually what is happening here. So for example, single cells in the presence of the vemurafenib, along the trajectory of ERK activity, we see pulses of ERK activation. So, and at the same time, because we have the ability to track individual cells, we can identify the relationships, genetic relationships, for example, between one another, whether the cells are basically genetically similar because they are daughter cells or sister cells or cells that are from different lineages. So what I'm showing here is a lineage tracking plot for single cells in DMSO and single cells in vemurafenib. And I'm highlighting one pedigree of cells that are basically highlighted by the dark blue color. The division events are shown by increases in Geminin intensity, and the ERK activity is high. In another case, in the presence of the vemurafenib, I'm showing one pedigree that is highlighted by the dark brown color. In that case, you see that at about the same time of cell division, we see reactivation of ERK signaling. So this is one of those cases that we can hypothesize that somehow the melanoma cell is escaping the effect of drug by reactivating the ERK signaling, and that's how the cell division happens. But there are a variety of cases actually that are somehow genetically similar because they're cousin cells, so it is less likely for genetic mutations to happen there. But you see that the cell divides after about 70 to 80 hours of exposure to drug without any sign of ERK reactivation. So this suggests that a combination of ERK-dependent and ERK-independent mechanisms might be involved in drug resistance and tolerance of drug and pushing of cells to a proliferative state. So to better understand what mechanisms those ERK-independent mechanisms might be, we have performed RNA sequencing in cells in the presence of drug and in the absence of drug a couple of years ago. And through RNA sequencing and gene enrichment analysis, we've found processes associated with neural system development and neural differentiation to be upregulated in cells that tolerate the effect of drug and go back to a slowly cycling state. And this was not very unexpected because we know that melanocytic cells are developing from neural crest in the origin. And what's happening here is that we can probably justify that by inhibiting the MAP kinase pathway or BRAF and MEK inhibition, we're pushing cells back to a neural crest-like state, which expresses the neural crest growth factor receptor. So to further test this hypothesis and understand the dynamics of these in coordination with cell cycle progression, we co-stained cells with NGFR, the neural differentiation marker, and a proliferation marker Ki67 in the same cell line called 58. In the absence of drug, cells are proliferating, expressing a lot of Ki67, but not much NGFR. And 24 hours after treatment, we see that cells consistent with the live cell imaging begin to get arrested within the cell cycle, decreasing their Ki67 levels. And then about 72 hours later, those Ki67 low cells increase their NGFR levels, signs of neural differentiation. And then after 5 days, those NGFR high cells begin to progress through the cell cycle and increase their expression of Ki67. Interestingly, if you remove the drug from these cells, it takes them 9 days, about 9 to 10 days in the case of this cell line, to go back to their initial state. So this suggests some adaptive rewiring is happening, and these differences between differentiation states of melanoma cells and drug resistance is probably not due to stable genetic mechanisms. There's some adaptive rewiring and non-genetic mechanisms involved. To further prove this point, we sorted cells based on their NGFR expression levels after drug treatment in 2 categories of NGFR high and NGFR low cells. And then we measured independently their doubling time to confirm that the NGFR high cells are doubling with a slower rate compared to NGFR low cells. And also, we removed the drug from each of these subpopulations of cells independently and tracked their NGFR expression levels. And we found that within 9 days, these cells gradually change their behavior and acquire exactly the same state that they were initially in terms of NGFR expression. So this further confirms that some adaptive rewiring mechanism is involved that is associated with changes in differentiation state and drug resistance in melanoma cells that takes 9 days to revert back to their initial state. So understanding the origins of this is important, because by understanding the mechanisms that are involved in drug resistance in de-differentiated melanoma cells, we probably might be able to propose strategies, combinatorial drug treatment strategies that could overcome drug resistance or prevent it in the first place. So we decided to focus on this problem more systematically, but at the same time, we wanted to do it in a broader manner. So basically, we included another form of differentiation state that have been discussed in the literature significantly, and that is even a less differentiated state of melanoma cells that is called undifferentiated cells. And these cells are basically expressing a lot of receptor tyrosine kinases, including AXL, that are used as markers for this differentiation state. Looking at melanoma cell line panels and tumors from patients within the TCGA, we can basically categorize these tumors and cell lines in, from differentiation states from melanocytic to neural crest to undifferentiated. And what we observe typically is that the more cells go from melanocytic to undifferentiated, the more intrinsically resistant they become to drug treatment with MAP kinase inhibitors. So to perform a systematic exploration of how these differentiation states are associated with drug sensitivity at a single cell level, we designed a high throughput multiplex single cell imaging experiment in which we assay differentiation state markers systematically at a single cell level by immunostaining and by measuring levels of MITF, the melanocytic marker, NGFR, neural differentiation marker, AXL, the undifferentiated state marker across all the 16 melanoma cell lines and primary melanocytes that I showed, and treated them with either BRAFIMEK plus chromatin, or DMSO for periods from 0 to 120 hours. And basically by combining all of the data at a single cell level and integrating them into t-SNE plots, we can see a spectrum of changes in differentiation state coming from basically portions that are NGFR high, MITF high, and AXL high. And as you see here, cells, AXL high and MITF high cells are almost mutually exclusive. And the NGFR high cells lay somewhere between MITF high and AXL high cells, which is consistent with transition of cells from melanocytic to neural crest to AXL high during the, basically AXL high to neural crest to MITF high during the differentiation trajectory. So given that we have these single cell data, we can overlay single cell behaviors on these t-SNE maps to understand what happens in each individual cell line. So this is Colate58 cells that we talked about. Initially, in the absence of drug, they are NGFR low. And as we increase, as we expose them to drug treatment, what happens is that we see changes in differentiation state. They begin to move toward NGFR high state. And if you combine vemurafenib with trametinib, the NGFR high state is even achieved in a more efficient way. So it enhances the de-differentiation process. So we can do the same across all of the melanoma cell lines that we're studying. And we see a diversity of responses that are associated with highly heterogeneous behaviors in some cases, such as MAC-SF or Colate58, or in some cases, for example, in the case of fully undifferentiated cell lines, pretty much no big changes are happening. They remain drug resistant and they do not change their differentiation state behavior either. So then we asked, now that we have this behavior of heterogeneous behavior across melanoma cell lines, can we associate that with patterns of responses in MAP kinase signaling and cell proliferation? So we did a similar experimental design with multiplex imaging, but at this time we measured basically activity of ERK signaling, activity of basically SX kinase, which basically measurements of phospho-SX is a protein associated with survival, and cell cycle progression by measurements of Ki67, a proliferation marker. So we did all of these together again in the same melanoma cell lines, in the same treatments, combined all of the single cell data into t-SNE plots, and then looked into individual cell line behaviors to see what happens with responses to MAP kinase inhibitors. So in the case of Colate58, as you see here, in the absence of drug, cells are expressing a lot of ERK, phospho-SX, and they are proliferating. And then as you increase, as you expose cells to vemurafenib with time, cells begin to move toward phosphoric low, phospho-SX low, or Ki7 low state, but you see a lot of heterogeneous behaviors. And if you combine vemurafenib plus trametinib, an increased proportion of cells are going to phosphoric low state, phospho-SX low, and Ki7 low state, but still you see some heterogeneity. So we can do the same across different melanoma cell lines to see the general observation that these responses are very heterogeneous and different from one cell line to another. In some cases, you see pretty much good responses with combination of MAP kinase inhibitors, but in cases like, for example, LOX-IMBI that was undifferentiated and drug resistant, even combination of BRAF and MEK kinase inhibitors are not doing much. So the question was that how the patterns of heterogeneous behaviors in MAP kinase activity and differentiation states are related. So we performed a full combinatorial correlation analysis. And these three plots are the major interesting plots that we can get. And basically, the more cells are pushed toward the phosphoric low, phospho-SX low, Ki7 low cells, the lower the normalized growth rate would be. So, and that is expected. So basically, drug sensitivity is directly correlated with the ability of drugs to push cells to fully inhibited state. Interestingly, the fraction of axial high cells are anti-correlated with this fraction of fully inhibited cells. So basically, cells, the more they are in the undifferentiated state, the less our ability is to inhibit the MAP kinase signaling together. On the other hand, the NGFR high axial low cells, the neural crest-like cells, are the ones that basically survive the treatment. But in that case, the fraction of these cells are also correlated with the fraction of cells that are fully inhibited. So we hypothesize based on these behaviors that there are at least two possible modes of drug resistance that have been discussed in the literature, but we are summarizing it here. One is that in the undifferentiated state, axial high cells, the drug resistance is actually associated with incomplete or lack of inhibition of ERK signaling. But on the other hand, in the case of neural crest-like cells, NGFR high axial low cells, ERK signaling is being inhibited, but cells survive the treatment because of their reduced requirement for ERK signaling. So we wanted to see what are the ways that we can approach this problem, other vulnerabilities that are unique to axial high versus NGFR high axial low cells, that we can either help these cells inhibit ERK signaling or enhance the requirement for ERK signaling. So basically by combining them with MAP kinase inhibitor, we see a better response. So the approach that we took here was an epigenetic compound screen. And the reason for that was the behavior of cells are associated with changes in differentiation state that have been described to be epigenetically regulated. And the fact that these responses are basically associated with a variety of changes and not individual changes in signaling pathways, but many different signaling pathways based on what we understand from the literature. We thought chromatin level mechanisms might be involved. So basically we designed an epigenetic compound screen in which we exposed either of the two of our heterogeneous cell lines to 276 epigenetic modifying compounds individually or in combination, in sequential combination with vemurafenib and trametinib for a period of 5 days. And then we performed statistical analysis to identify those compounds, those epigenetic compounds that change the growth rate of either of these melanoma cell lines, either by themselves or in combination with the BRAFIMEK inhibitor. So we performed the statistical analysis in a way that any significant change in either of these cell lines, either individually or in combination with the BRAFIMEK inhibitor would be captured. So we can basically narrow down the list of our epigenetic compounds, and we have a significant group to perform a follow-up analysis. And out of these, about 58 compounds came out to be significant. Then we use these 58 compounds in a more systematic way, basically by exposing again the same cell lines and the same drugs and measuring differentiation state, proliferation markers, and phosphoric levels in our cells, and we integrated all of the data together. The reason we did this was that because we did not know really what these epigenetic inhibitors are doing beyond the nominal targets of these compounds based on their manufacturer information. So we wanted to see if we have multiple compounds that target the same epigenetic enzymes, are they doing similar things to the cells in terms of differentiation state changes, ERK activity, or proliferation? And interestingly, we found some patterns that show some consistent basically behaviors. For example, we identified inhibitors of KDM1A, inhibitors of catencurase FETBRO domains, HDAC, and RO kinases, and two classes of HDAC inhibitors that are somehow correlating with one another. And these data provided multiple directions for us to follow up. But what I want to talk about today is patterns of responses to two structurally distinct classes of histone demethylases, one the KDM1A inhibitor, and the other group is the Jomanji family of histone demethylases that they both came out as important targets. So we took compounds from these groups and we exposed the panel of 17 melanoma cell lines to either of them, and we measured normalized growth rate. And as you see here, the interesting observation that we found is that they are like among these two compounds and across the 17 melanoma cell lines that we studied, basically you can identify either of these compounds to be working in basically in those melanoma cell lines, either individually or both of them are going to have some partial effects. For example, the cell lines that are very sensitive to SP2509, an inhibitor of KDM1A, seem to be relatively resistant to JIB-04, but there are some cells that are partially sensitive to both of them. And we see that cells that are very resistant to SP2509 is partially sensitive to JIB-04. We also combine these with BRAF-MEK inhibitor, vemurafenib, and MEK inhibitors and use high single agent model to evaluate the benefit of drug combination. And we found that basically cell lines that are sensitive to JIB-04, they benefit from the combination of BRAF-MEK inhibition, but cells that are sensitive to SP2509 show inconclusive results. So basically, in most cases, they do not benefit from combination of the treatment. So these data together suggest that across the panels of melanoma cell lines that we looked into, they are either very well sensitive to SP2509 or pretty much sensitive to the combination of JIB-04 and BRAF-MEK inhibitor, which is interesting because by either of these two drugs, we can cover the whole spectrum of cell lines that we are studying here. So to show it more directly, these are examples of cell lines that are sensitive to JIB-04 and combination of it with BRAFIMEK inhibitor, but they are resistant to SP2509, as shown by the blue color. And these are the cell lines that are very sensitive to SP2509 as a single agent, but they're not much sensitive to JIB-04 or its combination with BRAFIMEK inhibitor. And interestingly, in primary melanocytes within the first 5 days at least, neither of these drugs are working. So the next question we asked was that, can we identify any relationship between differentiation state changes or the baseline state of the cells, for example, baseline differentiation state or MAP kinase activity, and the effect of these drugs? So we extended our analysis across 10 melanoma cell lines. And basically, we measured a variety of markers in NGFR, AXL, MITF, and SOXN as differentiation state markers, and the activity of ERK signaling by phosphoric A7 or phospho-SX in response to the most important groups of epigenetic compounds that we found through our screen. And then we used partial least square regression analysis to connect the input measurements, the baseline and treatment-induced changes in ERK activity, proliferation state or differentiation state, and variations in normalized growth rate, either individually or in combination with BRAFIMEK inhibitor. So what PLSR is doing here is transforming the multi-dimensional data, the 24-dimensional data in the signaling and phenotypic state, into basically a PLS component space that can capture statistically significant correlations. And we found that for the models that we developed for SP2509 and JIB-04 plus vemurafenib plus trametinib, we see predictive models. And these data are showing the measured responses versus predicted responses after cross-validation. We won out cross-validation, showing that our models are able to be trained across 10 cell lines and predict across 9 cell lines and predict the 10th cell line. Then to identify which of our measurements are actually doing this prediction, we looked into the variable importance in the projection scores. For SP2509, we found that baseline levels of AXL are very predictive. And for JIB-04 combination with vemurafenib and trametinib, we found that the neural crest-like phenotype, high expression of NGFR, low expression of AXL, is informative, and it's usually working in cell lines that are also relatively sensitive to BRAFIMEK inhibitor, but not fully, partially sensitive. So they inhibit their phosphoric A7 and phospho-SX, but JIB-04 can increase the efficacy of MAP kinase inhibitor significantly. So to show this at a single cell level, we again went back to our single cell data. So this is directly the single cell correlations, the fraction of AXL high cells that we showed that they're resistant intrinsically to MAP kinase inhibitor effect are associated with changes in normalized growth rate in response to SP2509 alone. And the fraction of the baseline fraction of NGFR high AXL low cells is correlated with responses to JIB-04 in combination with BRAFIMEK inhibitor. So these studies actually so far suggest that there are variability in differentiation state of melanoma cells, and two of the important variabilities are associated with drug resistance, at least the neural crest-like state and the undifferentiated state. They are associated with different modes of drug resistance. One is more associated with intrinsic drug resistance and lack of inhibition of MAP kinase signaling. And those cells are sensitive to SP2509 by itself without combination with BRAFIMEK inhibitor. And another mode of drug resistance, the neural crest-like cells are able to inhibit their MAP kinase levels in response to vemurafenib and trametinib, but they reduce their requirement for ERK signaling, so some of them survive. And combination of JIB-04 with those drugs can increase these sensitivities and lead to basically more efficacious responses. So then the other question that we asked was that whether exposing our cells to longer times of treatment with either of these compounds could be basically associated with improvements in responses, or do we achieve similar adaptive resistance phenomena that we see in MAP kinase inhibitors? So we exposed our cells, 3 of the very resistant melanoma cells, 1 AXL high and 2 NGFR high cell lines, to either of the drugs that were predicted to be working in these differentiation states. And as you see here, because the log2 normalized cell count is shown here, so it means that SP2509 and A2058 cells can reduce the number of cells by 1,000-fold in comparison with vemurafenib and vemurafenib plus trametinib. And in the other case, basically the JIB-04 also enhances these responses without any signs of adaptation, at least within the first 3 weeks of treatment. So this study motivated us for performing additional follow-up studies in mice, but these experiments were interrupted because of the COVID, and we are getting gradually started back to the lab to basically performing these animal experiments. But we are generally excited about these studies because the SP2509, the KDM1A inhibitor that we're talking about here, is actually under clinical development for another type of cancer, the Ewing sarcoma, and it has been approved for phase 1 studies. So basically, there is opportunity here that if we find out that the axial high melanoma cell lines, that melanoma cells that are highly resistant to MAP kinase inhibitor are sensitive to these treatments, that would be a potential strategy to follow. On the other hand, the JIB-04 is an investigational tool compound, so we don't see that the compound is going to be used in the future, but it is motivating us for further development of compounds that are doing similar things by inhibition of Jomanje family of histone demetallases and their use to increase the efficacy of MAP kinase inhibitors in neural crest-like cells. So the last slide I want to show here is a little bit basically going into the mechanisms of the effect of these compounds. So we have only confirmed the effect of these compounds on inhibition of histone demetallases. Our next step would be to understand the actual mechanisms that kill the cells or overcome or help them overcome resistance to MAP kinase inhibitors. In the case of JIB-04, this compound has been developed by our collaborator in UT Southwestern lab of Elizabeth Martinez. And it is a pan-Jomanje KDM inhibitor that inhibits KDM4A, 4B, 5A, and 5B. But we wanted to know which one of the Jomanje histone demetallases are actually responsible for survival of neural crest melanoma cells. So basically, we performed siRNA knockdown experiments focusing on either of these 4 histone demetallases, and we found that KDM4B is KDM4B knockdown by siRNA could phenocopy the effect of JIB-04. So we don't have any selective KDM4B inhibitor available, but these results motivate us for potential development of such inhibitors, hopefully in the field. On the other hand, we also confirmed that in the case of the KDM1A inhibitor sensitive cell lines, such as the axial high A2058 cells, we don't see much effect for the inhibition of KDM4A, 4B, 5A, or 5B by siRNA, but knocking down KDM1A is pretty effective in killing these melanoma cells. So at least so far, based on these studies, what we are suggesting here is that these studies show that by epigenetic modulation, we can extend the BRAF oncogene addiction paradigm. And because of cases like KDM1A inhibition efficacy in axial high cells, we can also show that it's not just inhibition of the oncogene that can increase or help the tumor cell or the patient to overcome tumors, such as the axial high BRAF-mutant melanoma cells, but maybe the presence of the BRAF mutation in certain epigenetic states or differentiation states can basically provide additional vulnerabilities, additional epigenetic vulnerabilities, in this case, KDM1A state that could be targeted. And in other cases, basically epigenetic modulation can enhance sensitivity to BRAF inhibition. So both of these scenarios is actually considerable here and could be extended to other types of cancer as well. Another conclusion that I wanna make here is that our studies is a proof of concept that shows high throughput multiplex imaging to characterize heterogeneity with data modeling can be a powerful approach to further understand the origins of heterogeneity in drug response and develop potentially useful combinations to overcome drug resistance by studies that can capture a variety of heterogeneities and spectrums of heterogeneity at the same time. With that, I want to thank everyone who have contributed to this work, especially my lab members again. and I'm happy to take any questions.
Thank you so
Thank
much.
you so much.
This would be, yeah, this would be the time where you would have lots of applause. Thanks for that. We have a question in the chat box from Philip Stoffer. He says, did pulses of ARK activity correlate with cell fate, death over division? And have you looked at any genes downstream of the transcription factors looked at in this talk?
Yeah, good question. Yes. So we haven't done a full spectrum, like full analysis on the ERK pulses and their correlation with drug resistance, but there is another really good paper from the Sorger lab that was recently published a couple of months ago. And in that study, they have extensively investigated the origins of these ERK pulses and how they are associated with the emergence of resistance to both BRAF inhibitor and MEK inhibitor. So we just used it as a proof of concept to show that not just that is responsible for emergence of drug resistance, but in many cases, these ERK pulses are really important. For the second question, can you repeat it again?
Absolutely. It said, have you looked at any genes downstream of the transcription factors looked at in this talk?
So that is what, so in terms, if you mean the histone demethylases that we are targeting, yes. So we are beginning to look into that very extensively through RNA sequencing, epigenomic analysis, and proteomics to capture what is actually changing by inhibiting either of these. Another way that we're going to basically extend more from the bioinformatics point of view is basically we want to understand what transcription factors are involved in controlling the gene expression programs that are affected by these epigenetic programs, because it is very likely that ultimately the drug targets that we're going to propose as possible solutions are not going to be these epigenetic targets that we're using for screen. Unfortunately, we don't have many successful examples of epigenetic drugs to be useful in the clinic. So we are at this point using them as investigational tools that can help us understand what pathways, what other genes are really important, and then focus on those as potential targets. So that is definitely a future direction for us.
As a follow-up to that, can you speculate on how or why you think the cells might be dying off in response to these epigenetic modifiers? I mean, it seems like they're targeting a very specific state, but how do you think that might happen?
Yeah, so I think we have started with some very preliminary data by proteomics of cells after treatment with SP2509, for example, the KDM1A, and it looks, we see signs of metabolic changes. So it is possible, because many of these epigenetic molecules are actually associated with the metabolic state of the cell very well. So it is possible that the metabolic state is controlled and we're targeting those. We also see significant correlations with pathways such as cell adhesion processes and things like that. But definitely we need more data across a larger panel of cells and more extensive bioinformatics analysis to narrow down the options, because many of these have been discussed in in the literature in the past, but we have the same situation of not being able to target every type of tumor with individual pathway inhibitors. So we think we are missing something that hopefully by using these epigenetic inhibitors across a more diverse panel of cell lines, we probably might be able to find better answers.
Nice. So there's a question by Mike Freeman, but I'm going to see if I can get him to ask the question. Mike, you should be able to.
Can you hear me?
Yeah.
Yes.
All right, perfect. So on your multiplex imaging, what are your programs used to quantify individual cells?
Yes. So we have developed a pipeline of combining different things at the same time, depending on their ability to do things. So we use a combination of ImageJ, Cell Profiler, and MATLAB, depending on whatever stage they are doing a better job at. But basically, the majority of work is done by Cell Profiler.
Okay, thank you.
Sure.
We have a question from an anonymous attendee. When you mentioned metabolic reprogramming, do you have an idea on which metabolic pathways are getting activated, and are they cell line specific?
There's a good question. So I cannot have a definitive answer, but basically early signs are showing basically the KDM1A inhibition to be blocking oxidative phosphorylation in melanoma cells, which have been previously associated mostly with more differentiated melanoma cells, but we are seeing it actually happening in undifferentiated cells. So this is again in the line with future direction. So hopefully we will have more definitive answers, better answers next time.
Thanks.
So I had another question for you myself. It was really cool to see the heterogeneity of movement toward these different states, the differentiation states that you're just defining with the NGF, AXL, and MID-F.
Yes.
Do you think by increasing the concentration, you would reduce the heterogeneity, the concentration of the drugs? So they're at relatively low concentrations or intermediate concentrations, I guess. If you just increase that, would you expect the heterogeneity to go down and they'd be pushed even stronger toward a specific state?
The concentration of MAP kinase inhibitors or epigenetic drugs?
Yeah, any or all, whatever drugs you're talking about. So
that is actually a really good question. So it's very interesting. It is very cell line dependent. So we looked into changes in the heterogeneity of cells by statistical metrics, and it is really like difficult. Like in the case of sensitive cells, it is difficult to make a rule what's happening. But in the case of more sensitive cells, typically drug concentration is inducing heterogeneities, reducing the heterogeneities. But in the case of fully drug resistant cells, it is pretty much inducing more heterogeneity in terms of differentiation state. So we think like, because it is possible that it's a like full spectrum of behavior, but maybe sensitive cell lines are covering portion of this behavior and resistant cells of another. So basically we see that as we increase drugs, some are increasing heterogeneity, some are decreasing. But at least based on what like the single dose and comparison across different cell lines, we don't have any like consistent pattern to say like what is the actual heterogeneity. Is there like significant changes in one direction for heterogeneity changes? But that is a good question. Because of that, we pretty much focus all of our analysis on the baseline variability in melanoma cells. And it's interesting that even without considering what happens with drug MAP kinase inhibitor treatment, the baseline variability in differentiation state, which is reversible. If you, for example, sort NGFR high and NGFR low cells, they're going to go back to their baseline state. So that seems to be the richest, having the richest information for predicting what the epigenetic inhibitor they're sensitive to.
Yeah, so that's a fantastic, fascinating take on it. It's kind of consistent with the way we're viewing it as well. But what we're curious about is how any of those balances of heterogeneity are maintained within a population of cells. What makes a particular cell type result in that particular distribution of cell types?
Yes, that is a really important question. Yeah.
Right. Any other questions? Please drop them in, or you can raise your hand and you can speak if you like. If not, in a few minutes, we're going to transition to our small discussion. I'll post the link.
Just got it.
Oh, awesome. Thank you, Darren. So yeah, so we'll see you guys over there in just a few minutes.
So yeah, we would like to invite all students and postdocs interested in continuing to have open discussion with Dr. Fallahi Sichani. Please follow us over at the Zoom link that I just posted, and we will start up in about 5 minutes. Do it at 1 o'clock. Give you a break.
Thanks so much.
Thank you.
Thank you.
I'll see you soon.